Good afternoon, everybody. Those that are here live and those that are connected through streaming. It is my pleasure today to introduce our guest speaker. She is Anne Charmantier, and now comes some French, which is not sure I will be able to perfectly pronounce, but she's Directrice de Recherche Première Classe at CNRS. That's the end of the French. Comes more. In the Centre d'Ecologie Fonctionnelle et Évolutive at Montpellier. Finish with the French. She completed her PhD in Montpellier in 2003 and was postdoctoral research at the University of Oxford in between 2004 and 2006, and then returned to CNRS, started CNRS, and went through all the stages till the highest that I mentioned just before. And in 2011, she got the bronze medal of the CNRS, and in 2013, she obtained an ERC starting grant. And she was past president of the Society for the Study of Evolution, the editors of Evolution, chair of the European Society for Evolutionary Biology Equal Opportunity Committee, is member of the editorial board of Evolution Letters, if you happen to submit something. Now you have someone here to talk about. And also member of the organizing committee of a Woman in Science mentoring program. And as I read in the webpage, she defines herself as an evolutionary ecologist, particularly interested in how animal populations can adapt to rapidly changing environments. And I think she's going to talk about that today. And the title is there, so I don't need to repeat it. So please go ahead.
Oh, so I don't need to be near, I can walk around. I forgot I had my microphone. Thank you very much for this introduction. First of all, I want to thank Melinda for inviting me to give this seminar, and also Melinda and Rob for organizing the whole day and weekend. I'm really, really glad to be here in Madrid. It's my first time, and the museum is very impressive. It's really been a joy to discover the museum and chat with several people this morning, and I hope we'll have more chats afterwards. So I'm going to tell a story about natural selection, adaptation, and maladaptation in a particular environment, which is the urban environment. And in particular, I'm going to tell a story about one model species, which I've been studying for several decades now, the great tits. But before talking about the urban environment, I want to put you in the position I was when I started my career. So when I was doing my PhD in the early 2000s, this is where I started chasing blue tits. So we are here, let me give you the full atmosphere. Fango Valley, which is a UNESCO Man Biosphere Reserve in Corsica, in the northwest of Corsica. And it's here in this forest that Jacques Blondel set up the first nest box in 1975. And so we're celebrating this year the 50th year of this long-term monitoring of blue tits and great tits. And as you will see, the project has taken a breath of its own and encompasses many different avenues of research. My own interest in these species, the blue tits and great tits, is that I have explored questions around evolutionary and plastic responses of these two species, in particular in the context of rapid environmental change. And so we've been studying these past two decades in particular, the processes that underlie bird adaptation to climate change. So this is a topic that I'm not going to discuss today, but it has really taken a lot of my time in the last 2 decades. And the idea is that we use long-term data sets that are collected in these species that breed in nest boxes. So as many of you experienced in their everyday life, I'm trying to figure out, yeah. This is our main research tool here, a nest box in another valley in Corsica. And so we use these long-term data sets to optimize the way that we estimate natural selection, plasticity, and heritability by modeling the processes that underlie these factors. For instance, we try and understand gene flow between population, genetic drift, and genetic and epigenetic mutations. And finally, we're particularly interested in how the environment in all its complexity can influence these parameters. In particular, the fact that the environment is often fragmented, heterogeneous, and all the environmental stressors in which I will place climate change. Now, a complete change of sceneries, even though it's the approach that I've presented here is pretty much remains the same, but the scenery here is completely different. So this is a different atmosphere, different kind of stressors. I'm going to spare you the sound, which I recorded on the side of the tramway track in Montpellier. So this is my hometown of Montpellier in the south of France. Oops. And if I go back to this general framework, what we have gradually realized in the last 1 or 2 decades is that when one is interested in rapid evolution and rapid plastic responses, there is one landscape where we kind of expect that evolution should be prevalent, and this is cities. Why is that? Well, because cities are a concentration of characteristics that are likely to trigger many evolutionary and ecological responses. Cities are highly fragmented, they're highly heterogeneous, and they have all these environmental stressors. So you know about the urban heat island effect, the fact that it's warmer in cities. There's air pollution, water pollution, disturbance by pedestrians and their pets, by their cars, and then light and sound pollution. All these creates an environment that for many species novel in evolutionary timescales, and it's highly probable that it creates, that it affects many of the parameters and processes that we're observing here. Okay, so here is an outline of what my talk will be about. In the first section, I will tell you about what we know about natural selection in the urban context. In the second main part, I will tell you the story about great tits in this urban context, and a short section at the end will be about how we increasingly integrate the social factor in our eco-evolutionary approach. Okay, so what do we know about natural selection in cities? Well, in the framework that I presented, studying eco-evolutionary processes in the urban context, there is one consensus that emerges in the literature, and that's the fact that it's highly probable that natural and even sexual selection are very different in cities compared to what they are outside cities. Now, if we take a step back, I want to give you an overview here of what the literature has said about this issue of natural selection in urban context. And before urban evolutionary ecology existed, there's a field called urban ecology that is actually a long-lasting field. It's more than 50 years old. And when you browse through the literature, so here are some of the textbooks that were published from, I'd say, one of the first in 1977 to one of the latest one. If you look about the content in these books, they tell us stories about how cities influence species diversity, community composition, ecosystem functioning, and interactions between different species, especially people and nature and non-human species, and also how cities can impact biodiversity with often conservation projects. Now, what about the matter at hand for me, which is natural selection? Well, natural selection is often actually cited in these books, but in a way that I've used this quotation for Kevin Gaston's book, which is, I think, very representative of the way that these books present evolutionary contexts and in general, and natural selection in particular. And Gaston writes here, the urban environment represents a mosaic of novel ecosystems where natural selection operates differently compared to non-urban settings. So it's kind of a given in this literature that natural selection is different in cities. But in fact, my conviction, my belief when I started exploring this question was that I wasn't sure that we actually had the evidence, or that it was really estimated how natural selection was different in cities. So back to the general picture on evolutionary biology in urban environments. This is a field that is much younger than urban ecology. And I would say that it's around 2015 that started to appear review opinion papers about urban evolutionary ecology. This is a snapshot of some of the opinion and review papers that came out since then. I sometimes feel that in this that's more review opinion papers than there are empirical papers. That's my own feeling. But I think it's a sign of the enthusiasm that has emerged around this idea of studying evolution in urban areas. And one paper in particular caught my attention. This was published by Fougère and Henry in 2018. And this paper is in a larger context than just cities. It's about how human influences the strength of phenotypic selection, but not necessarily just in urban environments. And Fougère and Henry, they predict in this paper that we should see a shift towards stronger selection in areas that have high anthropogenic disturbance. Why is that? Well, because we expect that in these areas of high anthropogenic disturbance, there will be a shift in the adaptive landscape, in the optimal fitness. And so this necessarily should result in increased selection for the population to reach this new fitness optimum. But it is also for a different reason that we expect increased selection, and this is because increased anthropogenic disturbance will come with, overall, in most cases, a reduction in mean absolute fitness and an increase in the variance in relative fitness. So this is what you see in this graph here, that fitness will decline when you have high disturbance, and it comes with high variance in fitness, and so stronger selection strength. So what Fougère and Henry did is that they reviewed over 1,300 phenotypic selection coefficients that they gathered from 37 species. And they show in this review that there is indeed evidence for natural selection to be stronger in particular situations, which are fisheries, where there's especially strong selection for growing faster. But outside fisheries, in other disturbed areas, there's actually quite a lot of evidence for weaker selection. Now, I will give you a definition of weaker or relaxed selection as given here by Rodewell and Arsesi. So they've been working on the northern cardinal, where they found evidence for relaxed selection, and this is how they define it. Urban breeders might also be expected to experience relaxed selection pressures if these urban environments are buffered from deleterious effects of environmental variation experienced by rural breeders. So I'm giving a few examples here. For instance, in birds, it's often that urban environments provide new anthropogenic food resources, that there are less predators in cities, depending on the species we're looking at. And if we remove the cats from the equation, which is not always the case, and also warmer conditions, the urban heat island effect that I mentioned before, which can be beneficial, especially in winter. And so in the end, Fougère and Henry say, okay, well, maybe the situation is not that clear. Maybe there will be a lot of situations where anthropogenic disturbance will actually lead to relaxed, so disappearing selection rather than stronger selection. Okay, so in 2024, I gathered my group, and I was mandated to write a meta-analysis on the topic I wanted, and I said, okay, let's review natural selection in cities. And so we went through the literature. We thought it would take us a long time to retrieve all the selection coefficients we would find, and we wanted to classify them in evidence for weaker, stronger, or reversed selection, and of course also evidence for no difference in selection, which I haven't put up there because we didn't have a lot, probably because of publication biases. And what we found, so when we looked at all these selection gradients and differentials, we found only 8 studies in the urban context. So only 8 studies that have actually measured natural selection formally in urban and non-urban contexts. So we could not do a meta-analysis with 8 studies, but I'm giving you a kind of qualitative overview of these studies. So here are the studies that provide evidence for weaker, stronger, and reverse selection. And the dotted lines here are studies that didn't really measure a natural selection per se, as in a selection gradient, but they still have pretty good evidence for how natural selection is acting. And I'm going to give you 2 examples, one with a formal and one with a non-formal analysis of selection, so that you can understand what I'm talking about. So the first example is on the yellow jessamine, and this is a study that was conducted by Rebecca Irwin and colleagues in the area of Raleigh and Durham in North Carolina, for those that know the area, and cities around this area. And what Rebecca Irwin and her colleagues did is that they measured several traits on these plants, and especially around flowers. So they measured, here you have the traits. You don't need to understand the details, but they measured the floral display size, other parameters linked to flower size and shape, and also gelsomine as a resistant trait. And what you can see here are selection gradients. So I don't know if everyone in the room knows what a selection gradient is, but it's basically a statistical way to measure the link between fitness and the trait. And what you see here is that this link is very weak for most of the traits. It's close to 0. So it doesn't matter if you have a lot of, well, it matters a little bit for gelsomine, but all these PC1, PC2 have very weak selection acting upon them. But there is strong selection on floral display, and it's positive. So that means that the plants that have larger flowers have increased reproductive success. So they're selected by natural selection via reproduction. And what this study found is that this selection is stronger in the urban environment. So there's strong selection for having even larger floral displays, and the interpretation here is that it's probably linked to the decline in pollinators in cities in particular, which means that it's ever so important to have large flowers. Okay, so that was the first example so that you grab what kind of analysis we can do on natural selection. Now, the second example doesn't have a selection coefficient per se, but it's one of my favorite studies. To show how natural selection can act differently in cities. And these are studies that are done in the northeast of the United States, especially in this city of Syracuse here. And what the authors, so Cosentino and colleagues, have found is that they have been studying the European gray squirrel. And as it's rightly named, it's usually gray. Sorry, not the European, Eastern, it's not in Europe, Eastern gray squirrel. And so this squirrel is usually gray, but it's been increasingly observed that some of these squirrels can be black, so they have a melanic form. And this, if you are far away from a city, so here, for instance, 25 kilometers away from the city centers, the morph black is almost non-present at all. But the closer you get to the city center, the more frequent it becomes until half the population is actually black. So I think, you know, this is what we like about natural history when it's driven by an observation, something that we can't explain. And so what Cosentino and colleagues have done is a translocation experiment. So they collected gray and black squirrels, and they translocated them to rural and urban environments. And what you see in the rural, so outside cities, there is a clear advantage to be gray. You have here the mortality curve, so survival probability. And you can clearly see that because the black morph is more conspicuous, it's going to be, it has a higher predation rate, and so it has a much lower survival. What happens in the city is that, so the statistical analysis shows that there doesn't seem to be an advantage in being black, but it's very similar. So statistically, you survive just as well if you're gray or black. And this is because there's no hunting and there's much less predators of squirrels in the city. So this is a typical example of relaxed selection in the city where this lower predation and hunting makes it okay to be conspicuous. Okay, so this was just an introduction because it's like one of my interests to understand natural selection in cities, and one of the main message here is that basically we don't have enough data because there were only 8 studies that we can use. And so I encourage anyone that has data across urban gradients to have more formal analysis of natural selection so that we can sort out exactly what's happening. Okay, so now the story on gray tits. So here I'm going to follow a roadmap. We published this roadmap in a chapter of a book that was published in 2020 with my postdoc, Charles Perrier. And this roadmap is about studying adaptive evolution in urban environments, but really it's applicable to any kind of novel environment where you would find new phenotypes. So it starts at the top here, where usually the evolutionary ecologist starts questioning about evolution when we witness dimorphism or divergences in traits. So the leitmotif example here is a peppered moth melanic morphs. And so when we see such differences, as an evolutionary ecologist, for me, one of the first questions is to understand whether these differences are adaptive. And then we can also question whether these differences are of genetic or plastic origin. And this is done usually using experimental approaches. And then, well, there's the whole genomic section here, where we could also be interested in understanding how many genes are involved in these differences and where they are located. And you have different kind of approaches, basically depending on your budget, but also your model species. And so I'm going to use this roadmap here to present the results that we have gathered over the last 12 years on the study of gray tits in the urban environments. Okay. Oh, yeah. I first want to, so this is a project that started in 2011, so I'm still staying 12 years, but it's no longer 12 years, it's more like 14. And so this is an overview of the people that have been involved, especially Marcel Lambrecht and Arnaud Grégoire, who were there from the start of the project. And then a lot of the results are presented today are from the postdoc of Charles, PhD of Aude Kezerg and Megan Thompson. And these are the new people around now, PhD students and postdocs asking further questions. And also I want to mention the zoo of Montpellier. I will tell you about a little bit of a crazy experiment where we hand-raised chicks, and their help was really, really important to make this happen. Okay, so the Grey Tit project in Montpellier is a very basic monitoring based on nest boxes. So I think all of you are very familiar with this kind of monitoring. And here I'm giving a snapshot view of the forest of La Rouvière, which is around 30 kilometers from Montpellier. And this other snapshot here is about one of our city site. So the forest has been studied for blue tits and gray tits since 1991. It was mainly a blue tit study, in fact, even though the two species are present. And as I said, the city one started in 2011. And what we did is that we erected nest boxes in 8 different parts of the city, which have increasing urbanization. So you go from the zoo here that is really not urbanized at all to areas of the city where, well, basically when you're on top of the ladder and trying to tell the people at the bottom what is in the nest box, they can't hear you because there's so many trucks and stuff going past. So it's very urbanized. And what was important for us as well is that I don't think I have, maybe in this map, maybe you can see here, but in each of the urban areas, we attempted to have nest boxes both in parks, such as here. So every dot here is a nest box. You can't probably see the dots, but also nest boxes along roads, so that even within the sites, there is a gradient of urbanization. Okay, a few pictures, but I think all of you know about what monitoring a whole nesting breeding passerine is like, but basically we capture all the parents in the nest box when they feed their young, and every bird gets an individual ring. We measure classic morphological traits, we measure classic life history traits, such as the first egg laying date, number of eggs, number of nestlings, and number of hatchlings, fledglings. And also in our group, we're very interested in behaviors. I won't have a lot of time to give details on that. We can discuss this afterward if you're interested. But I will give you further on one example on the exploration score, which is done in this open field cage here. So I'm going to show you a video later on. So just so you understand the process, the bird is captured. And very quickly, we put the bird, even before its ring, in this little SAS room, so it rests here. And then it is released in this part of the open field cage here. And here is a camera and controlled light. And so we basically just film the bird exploring this novel environment during 4 minutes. And from that, we extract an exploration score, and you have a lot of variation between individuals. And this is repeatable variation across years. Okay, so let's start with the top of the roadmap, phenotypic investigation. Are gray tit in the city area different from gray tit in the forest area? The summary of what we have found is that every parameter that we have measured, every trait is different. So the bird and so some of these traits were not statistically different when I published the first paper, but now they're all really strongly significant. So the urban birds are smaller. They are lighter, they have shorter wing and tail lengths. This is something that we haven't fully explored why. So this is a call for, if some of you know about maneuverability and how differences in tail and wing length matter in the urban environment or in other environments, I'm really interested to hear about that. In terms of life history, the birds in the city, they lay around 4 days earlier in the city. They have around 2 eggs less in their clutches, and they have less hatchlings and fledglings. And then in terms of behavior, they have higher aggression scores. They are much faster explorers when placed in this open field cage, and they have faster breath rate when they are manipulated. And so obviously I'm presenting here results that are a contrast between forest and city, but the results are the same if we use the urban gradient, and if we do linear regressions, we find similar results across the whole gradient. And also what we've done now is that we have reconducted these type, for some of these traits that have been measured in other gradients. We had 13 different urban forest monitorings where we could revisit the same kind of analysis. And for instance, the fact that they're smaller is found in all the cities around Europe. And also, I should mention, because I'm really into quantitative genetics, but I'm not going to give you the details of these analyses, but for all of these traits where we could estimate heritability, these traits are heritable. So here we're in the presence of strong divergence between urban and forest birds for traits that we know are heritable. So obviously there is this question of whether this is adaptive, and this is linked to a kind of a microevolution and local adaptation to the city environment. And so to understand whether it's adaptive, Aude Kézergue, during her PhD, conducted selection analysis that I will present now. So as a reminder, birds are smaller in the city, and now the exercise is to understand whether being smaller in the city has an advantage in terms of natural selection. So I'm going to present here selection gradients. So for those of you that are not entirely familiar, a positive selection gradient here. So for instance, we have in green the forest birds, purple the urban birds, and the males are round. So here the forest males are selected to be larger because we're on tarsus length here. So if the selection gradient is positive, it means that there's a positive selection for this trait. And so what we found in this analysis is that overall morphology was not under very strong selection. So we have significant selection only for 2 types of traits, and that it concerns only forest males. So forest males are selected to be taller, and they're selected to be lighter, even though it's in the city that they are smaller. So we have no evidence that the city environment selects for this smaller morphology. In terms of laying date, I'm not going to show you the details because it would be too long. But as a reminder, birds lay earlier and smaller broods in the city. And well, actually, I am showing you the results, but they're not that complicated. So in the city, they're selected to lay earlier. So earlier means negative laying date. And yet the selection for laying earlier is true only in the forest and not in the city. And in the city, they lay smaller clutches, and yet they are selected to lay larger clutches. So all the evidence we have here of natural selection where we could measure it in terms of selection gradients, and it's similar for behaviors. This is where I'm not showing the details. All the behaviors that are associated with city life, such as being a fast explorers, are not associated with a reproductive advantage, but they're associated with lower survival. So overall, the picture here is that the observed phenotypic divergence doesn't seem to be due to an adaptive response, at least not in terms of natural selection as we can measure it nowadays. Of course, it's difficult to understand natural selection in the past, but from what we see here, some of these traits could actually be maladaptive because they're exactly contrary to what natural selection is favoring. I hope this is clear. By the way, I'm very happy to be interrupted for questions if there are burning questions. Okay, so the next approach is then to understand whether the divergences are from genetic or plastic origin. Since we have seen that these traits are not adaptive, we think that maybe most of them are just a plastic response of the constraints, but it is still possible that some of them are evolving in the city environment. And so this is where we thought, okay, crazy idea, let's do a common garden experiment, which I completely not recommend in a vertebrate, such as the great tit. So we collected eggs from the urban environment and the forest environment in Montpellier. And then we placed these eggs under foster parents for the incubation stage and the very early stages where chicks don't thermoregulate. They were all in the zoo, so all in the same environment, under foster parents, so not their original parents. And then as soon as thermoregulation was okay, then they were transferred to the nursery, and that's when we hand-fed these chicks from 7 a.m. to 9 p.m. for 3 months. This is where I would not recommend doing this. Luckily, in that year, we did that in 2022, all the chicks that had hatched, so we had 2 nests that were abandoned because of a horn nest and predators, but all the chicks that had hatched in the foster broods, they were all survived until the end. So in the end, we had 73 chicks that we phenotyped. So basically, we looked at their morphology. I'm not going to talk a lot about physiology and behavior and cognition, but we looked at all the traits that we had measured in the wild birds. Okay, and so the idea here is, of course, to check whether the differences that are seen in the wild are maintained in this common garden environment. So I'm going to show not all the results, but just 2 types of results, one on morphology and one on behavior. So here you have a reminder of the results in the wild. So this is the difference in body mass for the birds in the year when we raised the chicks. So in 2022, we found again that birds in the urban environment were lighter. This is something that we had found with the long-term data. What about the common garden birds? So there's quite an obvious thing happening here. First is that all the common garden birds are much lighter, right? And this is because they are raised in cages and then in aviaries, but they never fully fly, so they don't develop in this common garden. They don't develop their whole muscle structure, so they are overall lighter. But we also find that there's this persistent difference in that birds that are originating from urban eggs are still lighter than birds originating from forest eggs, even though they were fed exactly the same regime at libidum, like they have everything they need, perfect temperature, and they're still smaller. So this is kind of an incentive to think that there's possibly a genetic origin in this difference in size, since it's maintained in the common garden. Now you have here a video of this exploration score that I told you about. So this is what the camera sees from inside. And this bird is actually quite exploratory. So we measure the number of flights and hops, and we also measure the time it takes for the bird to explore the 3 perches and the entirety of the cage. And this bird will have done it in less than a minute, so it's actually quite quick. It's really exploring the environment. And so when we measure this in the wild, what we see is that urban birds are much faster explorers when placed in these cages. But if we do this on the common garden birds, what do we see? Well, this difference completely disappears. So birds originating from forest or city eggs, they explore the environment in the same way. Obviously, we only have 70 individuals for this test, because some of them were injured. So this leads to think that possibly the difference that we see in the wild may actually be a plastic response to the environment in the city and not a genetic response. Now I'm presenting, lastly in this part, a very recent results. So for some reason, we thought, these 2022 results are really cool. Let's redo the common garden. So we redid it in 2024, but we wanted to double the size, have 120 birds to hand feed, and we had eggs from Parus and Montpellier, and not just Montpellier. So they took the TGV train. It was a bit of a hassle. And so as a reminder, these are the results from 2022. So where birds are smaller, I showed you body mass, but it's the same. So the birds originating from eggs in the city are smaller than birds from the forest. And this is what we found in 2024. So I think this is a good lesson on why my colleague Samuel, with whom I did this experiment, told me, when something works, you should not redo it. You should just stick to the initial. So I won't have time to explain exactly why this happened, but the first thing is that we didn't have 120 birds, and this is because last year, 2024, was like the worst year ever in terms of reproduction. We had rain for 3 weeks, So all the chicks that hatched in the foster nests, most of them were abandoned by their parents because of the rain. So very few chicks actually reached the nursery stage. A lot of them didn't hatch, a lot of them didn't survive. And when they arrived at the nursery, they were in a dramatic health state. Already at 8, 9 days old, they were very, very skinny. So what we see is that overall, none of them reached the sizes that we had in 2022. But we do have this interesting result that it's actually the urban birds maybe that have coped better with restricted food and a very harsh condition. This is something that, so overall, the conclusion is that if the difference in size is due to genetic effects, this shows that there's probably strong genotype by environment effects. So when you're in good condition, the genotype shows that the forest birds are taller, but when you're in really bad conditions, stressful conditions with a lot of rain, well, the genotypes are not expressed in the same way. And it's possible that nestlings of city cope better with restricted resources, which is something that was tested by people in the Netherlands. And this paper just came out in Evolution Letters that actually really was testing this kind of pattern, where city birds cope better with very harsh conditions. Okay, the very last section of my talk is about genomics. I'm just going to give you an overview. With this study, only this study that we conducted in 3 different cities, Warsaw, Montpellier, and Barcelona, where all these cities have monitorings of grey tits in the city and the forest. And so we took blood samples from birds of these 3 paired forest city places. And we conducted rat sequencing to have genomic traces of selection in the city, and also RBS to look at the epigenomic marks. So this RDA redundancy analysis here, so for those of you that don't know, it's pretty much like a PCA. So every dot is a bird, and the closer 2 dots are, the closer their genomes are. This is a view of the rat sequencing data. And as you can see, the first thing that is really obvious is that there are clusterings via the cities, because here you have in orange the Barcelona birds, here the Warsaw birds, and here the Montpellier birds. I have the same symbols as before, where the males are round and the females are triangles. So the first axis of variation and the second are latitude and longitude, but the third axis of variation that comes out in the analysis is the urban-forest contrast. And you can kind of see it because, so the forest individuals are open symbols, and you can see that these open symbols in every city are clustered, and the full symbols are also clustered. So this contrast between being an urban bird or being a forest bird explains only 2.2% of the whole genomic variance in the rat sequencing SNPs. So it's not huge, but it does mean that there is a genetic structuration within the city, and that city birds have kind of a different genome, at least partly. This is a very similar type of view, but now for the epigenetic marks, so the RBS sequencing. And here I represent the first axis of variation, which is sex. So I had no clue about epigenetics when I started this project, and I didn't know that it's a very common result, even in humans, that males and females have very different methylation marks. So this is what we find here, that all the males are in the right-hand side of this first axis of variation, all the females on the left-hand side. And then the second axis of variation, not represented here, is the city. You can still see that they're clustered by colors. And the third, again, is this urban-forest contrast. But the percentage of variance explained in the methylation pattern is not stronger than the rat sequencing one. It's very similar, in fact. It's close to 2% of the variance in these methylation marks that are due to the urban-forest origin. And let me show you a different view. I promise this is the last result. So this is what is called a circus plot. I find it's almost a work of art. So the circus plot represents the grey tit genome. You have here all the macro chromosome 1, 2, 3, etc., that are quite big, and then the smaller one, and then you have a whole set of really teeny tiny chromosomes here. And in the center, you have birds from Barcelona, and then Montpellier and Warsaw. The red dots are areas of this genome that are hyper-methylated for urban birds. So urban birds have more methylation in this area. And the I was going to say yellow, getting tired, blue dots, are hypometilated regions that are under-metilated in urban areas. And so the two take-home messages from this circus plot is that first, there's no evidence for any parallelism, because if you take like a given area where urban birds are hypometilated in Warsaw, you will most probably not find it in the other cities. And this is true for most of the differentially metilated regions that we found. And the second thing is that we kind of expected hypermetilation in city. There are several studies showing that air pollution in particular can be associated with mutilation. And this is not what we find, because there are basically as many blue dots as there are red dots. So urban birds don't tend to be more hyper than hypometilated. Okay, so conclusions on these results. So I've showed you that basically on all the traits we've been measuring on these birds, there is strong phenotypic differentiation. Urban birds are different. This does not seem to be explained by the present natural selection as we can measure it. And we have seen from the Common Garden that some of these differences, however, could be due to evolution because they are maintained in the Common Garden. So there might be genetic effects or maybe very, very maternal effects in the egg investment, which is something that we will look into the future. And then finally, the genomic, epigenomic results show that there does seem to be an effect of urbanization on the genetic and epigenetic diversity, but it's largely polygenic. So we didn't find like a few set of genes associated with being an urban bird. And it's also, as we can tell from only 3 cities for now, largely non-parallel. So we don't see the same genes involved in urban adaptation. Okay, as kind of a conclusion, I want to end up on how we've been increasingly trying to include the human factor in our eco-evolutionary approaches. So obviously when you work in the urban environment, the human factor is important. And in fact, even when you're on the field, you talk to people much more when you're in the urban environment. We even had our ladder stolen at some point. So you get all these human adventures. In 2021, so this is a figure that was published in a Perspective article that was written by a whole collective of researchers, mainly North American, in 2021. And it depicts how social factors can influence the eco-evolutionary dynamics in cities. For instance, social processes such as political or economic drivers and decisions can impact the evolution by altering either the connectivity or the selection of plants in the city. And now what we've done in a project that I started 2 years ago is that we focus on the importance of culture and education, but also the feedback between our research and education, and how education can also feedback our own research. So this is a project that is funded by the National Agency for Research in France, the ANR. It's called Acacia Project. And in Acacia Project, we have 2 axes that are really about evolutionary ecology and a lot of physiology, adaptation to the urban heat island effect. And then the third axis is a educational science project. So this is not my field. This is led by Aurélie Swang, who's a researcher in educational science. And what we've done with Aurélie is that we've set up nest boxes. So we call them spy nest boxes because they have a camera inside and there's a solar panel so that the kids can basically see the whole breeding season, and they have projects with their teachers around what it's like to be a great tit during the breeding season. And what Aurélie and her team are trying to test is whether a science education based on observation and living the nature by observing what the birds are going through can reconnect the children with nature. And so this was actually a bigger project than I expected for several reasons. One of them is that you need to link different fields and like education authorities. It's very complex. But also what I realized, one of the challenge was to have the same semantics as Aurélie and like to understand each other. So the scientific process in educational science is very, very different from what we know in evolutionary ecology, but it was actually I learned a lot. So we did this last year and this year in schools in Montpellier and in Paris. And so you see here what's happening is that they have an app where all the different cameras are connected so that if the nest box is not occupied, they can see what's happening in the school next door. And there's actually, I think, a little video here, yeah. This is what they see basically at this time of the year, a great tit female feeding her nestlings. And the preliminary findings, we haven't published anything yet, but there's some really interesting stuff coming out of this project. In particular, the fact that the attention span of children towards nature has really increased throughout the year, probably thanks to this project. And I think that's an interesting conclusion that I hope will be formalized by all the analyses that are being done. And also it allows to introduce material, especially around ecology and evolution, that is not necessarily in the curriculum. So teachers are not familiar with these ecological facts, and I think even for the teachers it was very interesting. And then again, the challenges, but also rewards of interdisciplinarity, like true interdisciplinarity. You know, it's not like genomics and math and biology, they get along well, but educational sciences, another challenge. So these are a few examples of outputs from the schools. They all have very different projects. There was even like a theater and dance project. It was depending on the teacher, and it was pretty cool. Okay, I thank you for listening to me, and I think we still have time for questions. And again, I want to thank all my colleagues because this is very, very much a collaborative project that I've presented. Thank you.
Okay, thanks for the nice talk.
Thank you.
There's room for questions.
Hi, Anne. Thanks for a great talk. I really enjoyed it. So with regards to the DNA methylation results, I was wondering whether, I guess there's not because you would have mentioned it, but I was wondering whether you looked more closely at the sex chromosomes, because I know that there are some new studies or recent studies that found a differential DNA methylation within the chromosomes with age as well.
Okay,
in
so
birds,
yeah, the Z is here. The age effect, it would be impossible. I didn't mention the sample sizes, but they were very small. We had 60 individuals in total, so 20 from each, so 10 from a city and 10 from a forest from each. Unfortunately, again, mainly a problem of funding. So if anyone's interested, I have many, many samples that could be used for that. And the Z chromosome is here, yeah. And we didn't find any specific traces of selection associated to the Z or W chromosome, yeah.
Thanks.
Hi, thank you. Very interesting talk. I was wondering, you found a mismatch between the selection gradients that you found and the population differences between city and. How many years did you check that selection gradient? Because I wonder whether there could be some fluctuating selection. So you could have some years of extremely high selection that could explain patterns like that.
Yeah, that's a very good question. And we attempted to do annual selection gradients, but I don't think we had enough data. So I didn't put these up because I don't think they're very robust. Overall, when we did the analysis at that time period, we had 10 years of data. So obviously it depends as well on how many breeding per year, but we have about 50 breeding events per year across 10 years. I think at the time maybe the sample size wasn't enough, but yeah, in the future I'm really hoping to look at these annual fluctuations, because this is the feeling we get on the field. Like I told you about the 2022-2024 results. It's I think people find it annoying when there are very strong annual effects, but in fact I find it really interesting, and it comes over and over again, right? That when you look at a process very often, from one year to another, it changes a lot. And in the city in particular, heavy rains, or also I'm really interested in extreme climatic events. So for instance, in 2019, we had a heat wave of 47 degrees. And these are usually, events that happen only once every 10 or 15, or unfortunately more often than we would want nowadays. But yeah, it would be really interesting to sort out this annual variation and how important these extreme events are in the natural selection that we see. But we did an analysis like this on blue tits on lane dates, where we saw that extreme climatic events actually represent 20% of the total variance that we can see in a long-term data set of around 40 years. So even like 3 climatic events can explain 20% of the natural selection. So they are really important. But we haven't done this in the urban context yet. Yeah, thank you.
Thank you for your talk. It was very, very interesting. I was wondering if you could elaborate a bit on your understanding of weaker versus stronger selection, in the sense that the way at least I conceived it in my mind is that you may have weaker selection against predation, let's say, but in ecological space that just means that other traits are being selected for, let's say, fecundity or something else. So it's the way I understand it is not that it's necessarily weaker selection, it's just a stronger selection against something else, against another pressure. So how do you understand this weaker and stronger selection?
Thank you for this question, because I think, so my first approach was just to show the field that we can't just state in all these papers that natural selection is stronger in the city. It's just probably not true. For one of the reasons is exactly what you've said. Now, the way that I perceive natural selection is that we measure natural selection on a given trait. Now, you're talking about natural selection broadly on organisms. I don't know how you measure that. I would be interested. But it's certainly really important to have a multi-trait perspective, because you're completely right that if there is relaxed selection on your color, it doesn't mean that there's not stronger selection on your size or your exploration capacities to find food, things like that. So this is, of course, most probably what we will find in the end when we have enough data, is that does the city lead to stronger or weaker selection? Well, actually depends on the organism and on the trait, for sure. But this balance that you're talking about between weaker and stronger selection across traits, yeah, I have to think about it, how to measure that. Okay. But it's likely that it happens, yeah.
Thank you.
Thank you.
Hi, very nice talk. I was wondering, how close are these urban populations? Is there any gene flow with non-urban populations? Because that will impact
of course.
Selection?
Yeah. I didn't present that. It's something I should, just lack of time, but we did population structure analysis. And basically, in terms of geography, our city population is quite close, like it's less than 30 kilometers. But it's still, you have suburban areas between the true city where we're studying birds and the forest area. Some of these other sites that we've studied in Europe, the forest can be, like in Paris, it's 70 kilometers away from the city center. So that's variation across cities that is difficult to account for when you do meta-analysis. In terms of the true FST, so estimating the genetic structuration, we have an estimation of 1.8% of FST between the city birds and the forest birds. And to give an idea for those of you that maybe work on fish or other things where FSTs are usually very strong, 1.8% is actually almost what you have between, so for instance, for the subspecies of blue tits that we have in Corsica, it's 3% FST between the Corsican subspecies and the mainland. So actually, almost 2% of FST is actually quite strong. So overall, there is gene flow. Birds fly, and they can fly out of the city and out of the forest. But there is genetic structuration. That means there might be some local adaptation because gene flow is restricted. Sorry, that was a very long answer to a short question.
More questions?
Thank you, Anne, for the talk. It was very nice.
Thank you.
About this maternal effect that you mentioned, yeah, I think you have to look into it for sure, as we mentioned before the talk. Perhaps hormone-mediated maternal effects like testosterone, because you have smaller birds in the town, and they are perhaps fast explorers with faster developmental rate, so perhaps an early maternal effect could be influencing it.
Yeah.
So you have to take eggs. And
yeah. So thank you for stating this again, because I wrote here that this difference that is maintained in the common garden birds with the urban origin being smaller. We tend to say that it's a possible genetic origin, but it might be very early maternal investment in the eggs. So that's a possibility. Personally, I feel it's kind of unlikely because we don't have, so we did an analysis where we had a female as a random effect in all the models, and it doesn't explain any of the variants. So there doesn't seem to be variants across females that explain the body mass variants of the female of origin, so the true maternal mother, that explain any variance in the growth at the end. But it's still possible, that they have different investment in resources in the eggs, and that's something we haven't done yet. So I'm happy for any collaboration on this.
Following this question, I was wondering, again, or maybe it's a naive question, but whether there are some influence at the very early development of the birds by any alteration due to contamination or water or pollution or whatever, that might be interfering the early stages of development, and that might end in having smaller chicks, smaller adults.
Yeah, that's for sure. That's a possibility as well. So, in fact, what we should have done, and it's very complex to do, but Marcel Visser in the Netherlands did it, is to have a common garden across several generations, as you would do with the Drosophila or plant or whatever. You don't stop at F1, you go to F3 or something. So the birds don't breed in aviaries. So if you do that at every generation, you have to collect their eggs and then find new foster parents in the wild, and it's super heavy experiment. So we didn't do that, but for the things that you're telling me, this is what we should do. I mean, one thing is to analyze the eggs and see whether there are effects of pollution and things like that. But another is just to continue the common garden across several generations. I think I should switch to another model systems if I want to do that.
At some point.
Okay,
any more questions? No, so many thanks.
Thank you. Thanks, everyone.
Welcome.